Thank you. So good morning. Oh.
Good morning.
Oh, good. So anyways, thank you for the opportunity to talk about our work today. My name is Hisashi Umemori. I'm a professor of neurology and neurobiology at Boston Children's Hospital and Harvard Medical School. What we study is basically to understand how the brain is wired during development and how changes in wiring may lead to diseases such as PCDH19-related disorders. And today, I'll talk about work on PCD19-related disorder and specifically focusing on why this disease is female specific. Yes, it works out. Just, I don't know whether this might be better. Yeah, it's good.
Yeah, good.
And so, as you all know, PCDH19-related disorder. So I don't say epilepsy because there are many other phenotypes here. So that's why I said related disorders here. Is a disease associated with seizure clusters, cognitive impairment, and autism. And this disorder is caused by mutation in the PCDH19 protocarotene 19 gene on the X chromosome. But what's unique about this disease is that this is restricted to females or girls. Males with a mutation are normally unaffected. So this is very unique for a X-linked disorders because females have 2 X chromosomes while males only have 1. So that's why X-linked diseases normally affect males more severely. But this disease only affects females. So that was a question we wanted to understand. And that's what I will be talking about today. So PCD19 or protocarotene 19, and again, as you may know, this is a transmembrane protein. So this is a transmembrane domain, and this is inside of the cell, and this is outside of the cell. And this is mainly expressed in the brain, and this mediate calcium-dependent homophilic interactions, which means that this molecule binds to another PCH19, like this way. So here you can see like how PCH19 from one cell binds to another cell using extracellular domain, which is here, like this way. And that is how like a PCH19 connects 2 cells, or in the case of the brain, 2 neurons. And many mutations are found, but most mutations are found in this domain, meaning extracellular domain, where molecules used to interact with each other to connect cells. So, and then like many nonsense, or in the case of MSIT, in many cases, actually protein may not be expressed, but these mutations could be in the extracellular domain, suggest that like cell-cell interactions may be impaired in the patient. What you need to understand is this random X inactivation thing. Because the females have 2 X chromosomes, in each cell, only 1 X chromosome is used. So that's called random X inactivation. And in the case of normal, like a wild type people, human, then all cells would express PCD19, and in the case of brain, you will see like all PCD19 positive cells like this way. In female homo, where both X chromosomes are mutated, or in the case of male hemi, which only has X chromosome, and if PCD19 is mutated, then all cells would become basically PCH19 negative or mutations like this way. So in this case, there will be only PCH19 negative cells only here, like this way. But in the case of female head, this is what's happening. So in each cell, one of the X chromosomes will be used. If the chromosome X is normal, then PCH19 is expressed. But if the chromosome used is abnormal, then now that cell will lack PCH19. As a result, you get this random like expression of PCH19, and it appears this is a problem causing like epilepsy, autism, and cognitive impairment. And why this is happening is the question we were trying to understand. So first, we examined the expression patterns of PCDH19 proteins in the brain. And PCDH19 is indeed expressed by many brain regions, but two regions where PCDH19 is highly expressed are the cortex and the hippocampus. Okay? But the expression patterns are a little different. In the case of cortex, expression is high earlier, like a P7, actually even earlier, but then eventually the expression decreases like this way. While in the case of hippocampus, almost no expression was seen in P means postnatal day 7, meaning these are mice. So these are like after birth. And then that's basically like a P28 is kind of adolescence in mice relevant to humans, by the way. And in the case of hippocampus, almost no expression immediately after birth, but then expression comes up in a region called muscle fibers around P28, which is kind of adolescence in humans. So so that means maybe like these two PCDH19 in the cortex versus hippocampus are involved in different disease phenotype. And I'll come back to the cortex a little later. And then first I'll talk about the hippocampus and what's happening in the hippocampus. So mostly fibers, which is located here. This is where two neurons called dentate gyrus neurons and CSC neurons, CS3 neurons connect with each other to form synapses. And synapses are the site of contact where information transferred from the dentate granular cells to CSC neurons. And the structure is like this way. So you can see the terminal of mostly fiber here. And these are synaptic proteins where with these proteins, it can transmit information to CS3. And that's how information transfer from the dentate gyrus to CS3 in the hippocampus. And as you know, the hippocampus is critical for learning and memory. If synapses are not working well, you may have problems with learning and so on and so on. And PCDH19 is indeed expressed at mostly fiber synapses, which is shown here by immunostaining, where these are markers for synapses shown here. And PCDH19 co-localizes with these markers, indicating that PCDH19 is localized at synapses. And by immunoelectron microscopy, where you can clearly see where the molecule is located, and you can see PCDH19 in the presynaptic side, which means mostly fiber terminals, or postsynaptic side, which is CS3. And you can sometimes see they're kind of aligned like this way, as if they're indeed binding to connect the pre and postsynaptic side of the neurons to connect or form synapses like this way. So to understand the role of PCDH19 in the brain and in the hippocampus, we generated a mouse model of PCDH19-related disorder. Here, basically, we just inactivated PCDH19 by deleting this exon. And these are just to show an animal is made. Like here, in the case of male hemi, where PCDH19 protein is not expressed, then you can see a dark PCDH19 expression, meaning PCDH19 protein is gone. In the case of female het, you see that the expression becomes half. But what you can see if we magnify here would be the random expression of PCDH19, meaning some cells, around 50% cells express PCDH19, and the other 50% will lack PCDH19 in this case. Okay? And then using these animal models, we can examine what's happening in the hippocampus. So first, we examine whether axons, meaning the connection from the dentate gyrus to CS3 will be impaired or intact here. And by using this staining, which labels axonal terminals of dentate granular cells, you can see axonal terminals going like this way, or in the case of female het, going like this way. And we can also label neurons to examine axonal projections, which are shown here by dye labeling. You can also see axonal terminals like this way. So these images can look exactly the same, meaning axonal targeting in female het is normal in this case. So meaning the axons from these neurons indeed go to CS3. So that part is not impaired. So we then examine synaptic connections in this region, which is shown here. So as I told you before, at the synapse, there are many, these are synaptic vesicles containing neurotransmitters, and neurotransmitter released from here and received by receptors here to send information. So if you look at synapses like this way, you can see those synaptic vesicles here and the receptors here, and these are important structures for synapses to be functional. And that's what we examined. And what you can see here is this is the wild-type synapse, where you can see synaptic vesicles that contains neurotransmitters that are here. And then these are receptors. These dark lines are basically receptors being clustered. Okay? You can see like a synapse like this way, but if you look at female head, you can see much fewer synaptic vesicles being present in the presynaptic terminal of mossy fiber. And that's quantified here. And this defect is specific to female head, because if you look at male hemi, then they're normal. So it appears that there are some synaptic changes in female head at mossy fiber synapses. We then performed electrophysiology, which is to basically examine the synaptic transmission between the mossy dentate granule cells and CS3 neurons. So we can basically record the response of CSC neurons to examine whether synaptic transmission is normal or not. And this is one way to examine synaptic transmission, which is called miniature EPSCs, which is basically a spontaneous release of neurotransmitters. So to transmit information from dentate cells to CS3. And what you can see is this like a frequency of synaptic release is decreased, which means that indeed some impairment from this side to this side in this case. And this is another way to examine synaptic function called paired pulse facilitation. This is like in the case of like a wild type, if you stimulate this side twice, then you can see an enhancement. But this enhancement is actually exaggerated in female head. Okay? This also suggests some synaptic changes in female head, and male hemi was totally normal. And again, this is female specific. And finally, in terms of synaptic physiology, this is called LTP, or long-term potentiation. So this phenomena is considered to be the basis for learning. So we stimulate the brain or the regions frequently, then actually the brain will recognize that might be an important information. Like when you're trying to learn, like you try to learn things multiple times, then the brain will consider that's an important information. That's how we can learn by increasing the efficiency of circuit or synaptic transmission, and that is called long-term potentiation. And that we can examine by electrophysiology. So if we stimulate the brain multiple times here, then the brain will consider that might be an important information. And as a result, there is a kind of facilitation or potentiation of synaptic transmission, which you can see like here, initially like this way, but after stimulation, then it increases like this way, so that synaptic transmission becomes more efficient. And that lasts for a long time. That's why this is considered to be the basis for learning and memory. And this happens in the wild type animals, but almost totally impaired in female head, which is shown here. So LTP is basically missing in female head. LTP is normal in male hemi. So if a PCDH19 is totally gone, actually LTP is normal. Just a mosaic expression PCDH19 causes a problem of LTP, which may be linked to learning problems in a female head patient. So now then we examined what kind of behavior might be affected due to changes in synaptic transmission. So this a dentate CS3 synapses or hippocampus in general is very important for learning and memory. but we also know what kind of specific behaviors are indeed important, involve these synapses. So here I'm listing the three behavioral tests. First one is called contextual fear conditioning. This is kind of if you know, like a Pavlovian learning, meaning for example, like a dogs, if dogs hear sound and get a food, then they can just hearing the sound, they start to show some joyful movement. That's the kind of fear. In this case, we use fear, meaning they avoid the fear. That's the kind of fear conditioning. This involves hippocampus, but this synapse is not important for this kind of fear conditioning learning. In contrast, these two learning paradigm requires these synapses, which I'll explain next. So the first behavioral test is called contextual fear conditioning. This is basically for the animal to learn or associate cues or environment to the shock or fear. Okay, so what we do here is that we place an animal in this box here, and this is the box here. That's the environment, and then with a tone, and we give a shock to the mouse. Then the mouse will remember this environment, and tone will be associated with this shock, and as a result, animal will show a freezing here. As soon as it shows here's the tone and if placed in the box, animal will expect some shock to come. That's why animals will freeze. So, and then this is how animal can learn or associate tone and environment to the shock. Okay, and as a result, if we return this animal to this context, then even without a shock, this animal remembers this environment. That's why this animal shows freezing. Okay? And on the third day, now this animal is placed in a different environment. So at the beginning, this animal does not show any freezing response. But then when we play a tone, then now this animal recalls this memory. That's why this animal shows freezing. So this is how we test the learning of these animals in this paradigm. And this does not require multi-fiber synaptic function. And indeed, if we test female head or male hemi, we did not see any differences or changes using this learning paradigm. So with the shock, freezing responses increases, which was normal in female head. And when on the second day returned to this environment, animal showed freezing in the same way to the wild type animals. And when the tone is played, so in the beginning, no freezing, but after the tone was played, show freezing. Similarly, between wild type and female head, so did male hemi, like this way. Okay, so this one was not impaired, which is consistent with the fact that this does not require multi-fiber synaptic function. The second test is called pattern completion. So this is an ability for us to combine two information together to complete the pattern. For example, if you see someone coming from far away, it's your friend, and even if that image is so big, you still can recognize that's your friend. That is because that very fuzzy image can be combined with your memory, through pattern completion, to create a better image to recognize that is your friend. So that ability is called pattern completion. Okay? This ability requires multi-fiber synaptic function. And to test this with animals, we do this. So this is one group, and this is another group. In this group, animal is placed on day one, nothing, meaning basically stayed in home cage. And on the second day, animal is placed in this cage and given a shock immediately, and then return to home cage. Because this was very short period of time, animal cannot really remember this environment clearly. And that's why on day 3, when this animal returns to this cage, animal does not show strong freezing response, just weak freezing response, because this memory was very vague. Okay? Now, in the second group, here, the animal on day 1 first placed in this environment and does nothing. No shock is given, so that this animal can explore this environment, to remember this environment. And then day 2, same thing, just immediate short shock training here, and then can return to the home cage. But in this case, this animal already experienced this environment. So that's why they can associate these two, like memory, through pattern completion to create a better memory, so that animal, when returned to this case later, will show strong freezing. Okay, that's pattern completion. Is clear? Good. Yeah. So this is how we test pattern completion ability using animals. And this, so like in the case of wild type animals, with pre-exposure, we see like a more freezing, meaning these animals remember or like combined 2 images to create a better memory. And this ability was missing from female head, and no change with pre-exposure. And this is just a time course. So pre-exposure, you see differences in terms of freezing responses. Female head didn't really show any differences between no pre and pre-exposure, suggesting like a pattern completion ability is impaired. This is specific to female head because male hemi was totally normal. So male, even without PCDH19, shows normal pattern completion ability like this way. Okay. So again, this is consistent with the functional mossy fiber synapses and specific to female head. The third behavior is called pattern separation. So this is to basically separate 2 similar images from each other. So initially, if you look at 2 similar images, you can't really distinguish between these 2, but as you look them carefully, you can eventually separate 2 similar images. That's pattern separation. Okay, and then here with animals, this is what we do. We prepare 2 kind of similar, meaning like the shape is same, and basically everything is same except for 1 panel here. So in this case, this panel is basically just plain here, but this panel has a stripe here. Okay, with these 2 environment, and only A is associated with shock, and that's what we do. And so this is the training. So in A, we give shock so that this environment is associated with shock, so that animals show freezing responses when placed in this environment. Then from day 4, we place this animal into B and A alternating. And at the beginning, because these 2 environment or boxes are very similar, that's why animals show freezing responses in both A and B. But whenever animals are placed in A, shock is given. And eventually, animal can't distinguish between A and B. And as a result, they show through pattern separation, weak freezing responses in B and strong freezing responses in A. So that's pattern separation. And this ability also requires mossy fiber synaptic functions. Okay? And when we tested our animal models, and this is what we saw. So in the case of wild type animals, at the beginning, they couldn't really separate these 2 images, but eventually they could. So in the case of female head, this initial difference was not different even later. And this is a learning index where wild type learned well to separate 2 closely related images, but female head did not. Male hemi was normal. They can separate these 2 closely related images. So again, this is consistent with mossy fiber function impairment in female head. And again, this is really specific to female head. So together, our animals indeed showed female head specific phenotype where they have PCDH19 mosaic expression with dysregulation of mossy fiber synapses, like a long-term potentiation, and as a result, cognitive impairment, including pattern completion, pattern separation. So our animal models indeed show female-specific phenotype like this way. Now, of course, the question is why? this only happens in female head? So that's the question. So as I told you, PCA19 is a protein that binds with each other like this way. Okay? So, and if that is the case in the wild type, in a normal situation, do you want to hide it?
Blocking your slides. No, it's good. Why don't we just like you wanted to do initially?
Yeah.
Better, right?
Yeah, better. Probably better. Yeah.
Good.
Yeah, for you guys. Yeah. It's fine or better. It comes back. Yeah. Yeah. That's so. So PCA19 is a homophilic binding protein. So if that's the case, this might be what you expect. So here in the wild type, like PCA19 binds with each other to connect two neurons to establish synapses like this way, sending signals downstream. But if this is the case, in the case of in the case of male hemi, because all neurons lack PCA19, this should be the result, meaning no binding between these two neurons, so no signaling, so synapses should be impaired in this case. Okay, and female head-wise, it should be also impaired because there could be a mosaic, meaning that some cells express PCA19, some cells don't, and if they meet, they cannot interact, so there should be impairment. But if this is the case, there should be impairment in male hemi as well. But this is not the case. So that's why this is not this does not explain why the disease is female specific. So meaning we need to bring in additional players to explain why this disease is female specific. And that's when we focus on another cell adhesion molecule called N-cadherin. So N-cadherin was proposed to bind in a cis manner, like this, cis meaning like a bind like this way, like from the same cell, they may bind like this way, right? And if they bind like this way, this may mask N-cadherin's function like this way. And if this is the case, this is what you expect in the wild type, where still PCA19 to 19 binds with each other like this way, and N-cadherin might be binding here, but this function, the function N-cadherin is masked. And as a result, they may recruit some signaling molecules downstream to establish synapses here. Okay? And signaling molecules, one of the signaling molecules is called beta-catenin, which are recruited to send signals to establish synapses like this way. Now, if this is the case in male hemi, this should happen. Okay? So here, now PCD19 is gone from all cells, but now N-cadherin is unmasked so that they can interact with each other like this way, so that they can establish connections. Okay, and that's how they can recruit beta-catenin, send signals, and still they can establish synapses. Okay? Now, some connections. No, probably connections. Yes, good. Now, in the case of female head, this should happen. Okay? So some cells still express PCD19, but some cells lack PCD19. And if this is the case, now they cannot interact, okay? Because there's a mismatch between this side and that side, so that there's no recruitment of beta-catenin, no signaling. That's how the synapses could be impaired. So this is what we wanted to test, okay? So first, we wanted to know if indeed N-cadherin and PCD19 interact with each other. And we did 3 experiments. This is called co-IP, like when you put down PCD19, whether N-cadherin is also put down. If it's put down, then they suggest that they interact. Indeed, you can see a band here, which means PCD19 and N-cadherin interact. And this is to show whether they really co-localize at synapses. And you can see PCD19 and N-cadherin located at both C fiber synapses together. like this way or like this way. And this is called a proximity ligation assay, where signals are observed only when these molecules are really closely related. And you can see signals here in the wild type, suggesting that these two molecules, meaning PCD19 and N-cadherin, interact with each other. We then tested if mismatch indeed impairs synaptic development. And for that, we used culture systems. It's a co-culture between neurons and HEC, meaning like just like a cell line, which is not a neuronal cell. And when we culture neurons, they send axon and dendrites processes. And when they meet, that's where synapses should be made. And indeed, in culture, you will see synapses where axon and dendrites contact like this way. Okay. And if you place a cell like HEC cells, which is not neuronal, onto these cultures, there's no synapses between HEC cells and neurons like this way. But if we express synaptogenic molecules in HEC cells, now you can see synapses formed between HEC cells and these neurons. Okay? This is the assay. And what we prepared in terms of neurons and HEC cells were like this way. Neurons are wild type, meaning they express both PCDH19 and N-cadherin here. And in HEC cells, we expressed both N-cadherin and PCDH19. In this case, basically, this is a matching condition, meaning like wild type conditions, okay? Or N-cadherin only, meaning this is a mismatch, like female het condition, okay? And examine whether synapses are made at the contact sites. Okay? And here's the result. So here we expressed both N-cadherin and PCDH19, and at the contact sites, so red dots are synapses. You can see synapses here. But when we expressed only N-cadherin here, then synapses are missing. So if there's a mismatch between PCDH19 and N-cadherin, then no synapses are made in this case. So it indeed suggests that mismatch indeed causes synaptic problems. And then finally, we tested if the signaling is impaired or N-cadherin dependent signaling is impaired in vivo. And with that, we examined whether beta-catenin, a signaling molecule, is recruited to the synapse. So you can see in the wild type, you can get a nice cluster of beta-catenin like this way or like this way, but you can see much smaller dots here, and then much fewer beta-catenin recruitment to the synapse in female head here. Male hemi was normal. It was able to recruit beta-catenin to form synapses. So it appears indeed that is a problem. So meaning in female head, there appears to be mismatch between PCD19 and N-cadherin, and this mismatch causes impairment of signal molecule recruitment and synaptic establishment. Now, if this is indeed the case, okay, if we overexpress N-cadherin from this side, for example, then it can restore this connection like this way, right? And so that this can recruit beta-catenin like this way to send or restore signaling to re-establish synapses. Okay? So that's what we tested next. So we expressed N-cadherin, so into the hippocampus from both pre and post-synaptic side or only from the pre-synaptic side, and we examined whether beta-catenin is recruited and whether LTP, like physiology, can be rescued. And indeed, we can rescue both of them. So in both cases, we were able to rescue beta-catenin clustering after N-cadherin overexpression. Beta-catenin was recruited like this way, like this. So that's before. You can see smaller, fewer dots here, and now you can see enhanced beta-catenin clustering here. And LTP, like a basis of learning memory, can be restored. That's a female het, but after N-cadherin was overexpressed, you can see LTPs restored in this case and as well in this case. So these results in this support this model where in female het there is a mismatch between PCD19 N-cadherin complex and N-cadherin, that is the cause of female-specific phenotype leading to synaptic defects, LTP, and cognitive impairment. So what we are hoping to do is to basically utilizing this mechanism, applying this mechanism to the treatment strategies, like either like re-expressing N-cadherin to female head, or unmasking N-cadherin from here, which may include inactivating PC19 or dissociating this interaction to restore NCAT interactions so that synaptic functions and behavioral impairment can be rescued. And that is what we are trying right now. Now, I will move a little bit, just spend a little time to talk about the cortex, cortical phenotype. So, so far I focused on the hippocampus, which is important for like a cognitive or the learning memory or like a phenotype in patients, but also PCD19 is highly expressed in the cortex. And as I said, expression in the cortex is way earlier than in the hippocampus. Meaning like in patients, like for example, like a seizure may come first, and later like a cognitive impairment or autistic behavior changes may follow. So the cortex may be like important for earlier disease phenotype. And so, and then that's what, and then like including seizure. So, and we wanted to know what's happening in the cortex. And in female head, one change that's clear was this abnormal cell aggregation in female head, which was first observed by Paul here. And where you can see like a PC19 positive and negative cells abnormally cluster to show stripe patterns like this way. And this was also observed with our animals, where we stained for PCD19, and you can see PCD19 positive cells here and negative cells are located here, dark, to show abnormal cell aggregations like this way. And we can also observe by labeling the two chromosomes with different colors. One chromosome is red, which is normal, and the other black chromosome lacks PCD19, and you can see stripe patterns of PCD19 positive cluster and PCD19 negative cluster. Okay? Where like these two cell types are segregated to show abnormal cell aggregation. Okay, so and we wanted to know like how it happens, or why, or how it happens, or when it happens. So just to tell you a little bit about cortical development wise, so in the cortex, the first cells here are called radial glial cells. Those are basically progenitor cells to produce many cells in the brain, in the cortex, and they start to divide from E meaning embryonic day 10, very early in brain development. And these cells produce neurons starting around E12, continuing until E15 in the mouse to produce neurons like this way, and they migrate to the cortical areas like this way to form layers. Okay? So that's when neuronal migration starts. Then later comes cells like glial cells. Those are the cells supporting the cell function like this way. So this is the kind of the development of the cortex in the mouse brain in terms of time course. So what we first wanted to know was when PCD19 is important and which cells, from which cells PCD19 is required for this abnormal cell aggregation phenotype. And for this, we generated conditional allele for PCD19. So this is an animal model, another animal model here. So in this case, one allele of X. So in the case of female head, one chromosome is normal. This is a normal X chromosome, but the other chromosome is modified like this way. So here in this allele, we have first exon here followed by EGFP, meaning green fluorescent protein. Right? And in this case, if this first exon is deleted through Cre-mediated recombination. So there's a Cre-an enzyme to kind of delete a certain region of the genome. And if Cre can delete this exon, then now this green fluorescent proteins will be expressed, and PCDH19 is inactivated. Okay? So meaning this is like a PCDH19 knocked allele, but in addition to knocking out PCDH19, now green fluorescent protein will be expressed, meaning if the cell uses this chromosome, then cells will lack PCDH19 and become green, so that we can know that green cells are when other cells lacking PCDH19. Black cells would express PCDH19. So that's how we designed this animal. And the Cre lines to inactivate an allele we used, I will just talk about these two Cre lines. First Cre line is called RBP4 Cre. In this case, Cre enzyme is expressed, start to express around E13.5 when neurons are being generated to migrate to the cortical areas. So if neurons are responsible and the migration are responsible, then we should expect to see changes or abnormal cell aggregation here. And this is very specific to pyramidal neurons. I mean, if neurons are responsible, then this line would also result in defects. And the other is called EMX1 Cre. This starts earlier, like even E9.5, even earlier, and also Cre expressed not only neurons, but also radial glial cells, meaning the progenitors of neurons, and so on and so on, and glial cells, which are later. And in the case of RBP4 Cre, actually, we did not see any abnormal cell aggregation. So you can see green cells are here and red cells, and not red, black cells are here, meaning green and black cells are intermingled, meaning we didn't see any abnormal cell aggregation when we use RBP4 Cre, meaning either like a deleting PCDH19 from neurons was not sufficient, or E13.5 was not early enough to show abnormal cell aggregation phenotype. In contrast, in the case of EMX1 Cre, we see this pattern, abnormal cell aggregation patterns like this way. Okay? So where green cells lack PCDH19 and black cells located here still retain PCDH19. So this may mean that PCDH19, like earlier expression PCDH19 is responsible for this phenotype, and or cells like radial glial cells may be critical for this phenotype. Okay. Now, I just got blowing up this EMX Cre. You can see like abnormal cell aggregation here, not only in layer 5, but also layer 2, 3 here. So all layers are affected here, which may mean that radial glial cells may be responsible for this phenotype. Okay. So what we are doing right now for this part is are shown here. So we are indeed trying to understand whether PCDH19 radial glial cells is important for this abnormal cell aggregation, meaning that's whether these progenitors or cells guiding neuronal migration may be responsible for this phenotype, and whether this abnormal cell aggregation leads to abnormal brain activity, like causing epileptic activity. And that's what we are also trying to look by performing in vivo live imaging of brain activity. So we can actually examine brain activity now, like from a mouse brain, from a living, behaving animals. And this is just one example. So that's just you can see brain activity like this. You can see a nice pattern of activity running through the brain on the surface, because you see, so this shows the pattern of activity or waves of activity running through the brain. And that's what you can see by performing in vivo live imaging. So you can just open the skull and then see brain activity. That's what we can do. And we are trying to see if these abnormal cell aggregation may cause disturbed brain activity, and that may be the basis of abnormal like a brain activity or leading to seizure. That's what we are trying to see here. And then we want to know whether PCA19 and cadherin interactions are also responsible for this abnormal cell aggregation and maybe abnormal brain activity, because N-cadherin is also known to be involved in cell migration. So we believe it's likely that this interaction is also responsible for this abnormal cell aggregation, and that is what we are testing right now. And finally, if this is the case, then we hope to target N-cadherin for the treatment of PCA19-related disorders. And by targeting N-cadherin, we showed that hippocampal phenotype can be rescued. And we hope we can also rescue the cortical phenotype so that this can be a more general treatment strategy. One treatment strategy is for PCD19-related disorders. And that's what we are hoping to do and trying to do. So this is just the final slide. So here are the people who contributed to the work I told you today. So now Hoshina is here or somewhere here, who mainly did this work, which with Joshua Beckert, who is here, looking at the eclipse a couple months ago. And then so he's a postdoc. He was an MD PhD student from Germany. And so these two are like main contributors to the work, with Erin, who is an electrophysiologist, Chloe and Miyuki Kana basically helped provided technical assistance with other people, of course, and the support came from PCA19 Alliance and NIH and others. So thank you very much for your attention.
Microphone over here.
That's it.
Very exciting work.
Oh, good.
And I'm wondering if you also already mapped down PCDH19 at females and with that recover function or not? And if not, why?
Yeah, so if we knock down PCA19 from head, that is a female homo, basically PCA19 is gone, then indeed most phenotypes disappear. So indeed that's indeed absolutely one very good strategy that many people are now trying to knock down PCA19 from female head to make them like female homo, and that's I think that's like most I would say popular strategies and nowadays happening right now. Thank you.
As usual. I was just working with your EMS mouse. Did you do any like the cortical morphometrics? Did you see any evidence of like malformations or any differences in structure?
So yeah, so we are doing that right now. So yeah, so we are trying to look at the morphology or in synaptic connections and perform morphological analysis, physiology, and so on. So yeah, so soon we should be able to update. Yeah.
Trying to reconcile the model I have. You showed with the mutated PCH19 no extracellular domain. If it's knocked out, but what happens if there's a small missense mutation on that? Doesn't it? Don't you still have an extracellular domain? It's just looks different, it's folded differently.
Yeah.
How does that?
Good. Yeah. So what we think is that if there's a mutation in the extracellular domain, that will affect cell interactions. So meaning they will not bind to the wild type PCA19. So that's basically the same thing. So if you just I don't know how far you can go back. So here, so if there's a mutation here, I mean, they say there's a PCA19 still being here, but if this is mutated, then they cannot bind. right? So then it's basically the same. So there's a mismatch between this side and this side, so that they cannot interact. So that's a good question. Yeah. So if like there are free and cadherin being present there, and which we think is the case, then that should be able to rescue. But that's a great question. Yeah. Yeah.
So wonderful talk, the fantastic data. But do you think it's always incoherent that might be at play here with predicate here 19? Do you imagine there might be other extracellular molecules that are replacing incoherent in this kind of interaction that you've already shown for tasks with emulsifiers in other parts of the brain and other circuitry? Could you speculate about that?
Like others, like PCDH.
Are there other molecules that replace the role of incoherent that you've shown in the emulsifier context in other neuronal contexts within the brain? Do you think there are other molecules that are acting in that synapse with predicate?
Yeah. I mean, that's a great possibility. Yeah. I mean, if that's the case, yeah, that's absolutely interesting. Yeah. Right now we don't know. We have no ideas, but that is a interesting point to like kind of suggest certain like a specificity in the brains. And yeah, absolutely a great question. Yeah.
Right. I'm curious if anything found in these models explain the clusters of seizures that kids with PCDH19 have versus other types of epilepsy.
So what we are hoping to know is whether like a cortical phenotype is indeed linked to seizures. And that's why we are trying to examine the brain activity using in vivo imaging. And if there is abnormal brain activity pattern in the brain, then that may cause epilepsy. And if that's the case, these like abnormal cell aggregation, abnormal like a connection to the cortex happening in the PCA19 female head brains is a basis of epileptic seizures. As opposed to like others, there are many other mechanisms. Like in the case of Dravet syndrome, it's channels. Of course, it's different. But we hope that abnormal cell aggregation may be linked to like abnormal brain activity causing seizures, and that's what we are trying to understand.
Hi, that was a really great talk. If you had to guess, what do you think is the purpose of this shift in localization from cortical to the CA1 hippocampus throughout development? And also, did you see increased immunofluorescence for PCDH19 in other regions of the cerebellum?
So PCDH19 is most abundant earlier during brain development. Mostly fibrous synapses are like just a little later. And that is maybe partly because dentate granule cells where they provide presynaptic neurons continue neurogenesis throughout life, meaning that they continue to divide even in adult. And that may be a reason that the PCD19 continues to express, and those synapses are always refreshed throughout life. And maybe for that, PCD19 may be playing a role. But the PCD19 does not disappear even in adults. They still express, and indeed many other regions express PCD19. And those other brain regions as well, we are indeed interested in understanding what PCD19 is doing.
Great questions.
Just so interesting, great talk. Do the rats have experienced seizures? I think it's no. Right?
So no spontaneous seizures. Yeah. So I mean, of course, we can induce seizures, but we need to do other more experiments to see whether seizure threshold is indeed different. depending on the experiment, we can actually see the threshold is a little different in female head. That's also we are trying. Yeah.
With trigger.
Yeah, with trigger, we need to induce seizures
So do
to
you
see
suspect
changes.
something else that also
Something
made
else?
a difference in PCDH19 case?
So I think that's the difference between humans and mouse. So many seizure models in mouse do not really show spontaneous seizures for some reason. Like not only PCD19, like for example, like we also study CDK5, but mouse models do not show spontaneous seizures. Probably partly because some like structural differences between mouse and humans. Like mouse brains are much more smooth. Like as opposed human brains are way more complex, and that's like among others. So just like for some reason, mice are more resistant to spontaneous seizures.
Okay, so I have a question. I have a son that's 5 that has PCDH19. Would this also apply to a mosaic male?
I see. So yes, yes, I think so. So if there's a mosaic pattern, then same pattern should be observed, meaning mismatch like this way. And I believe that's the basis of the disease phenotype.
Thank you.
So this might be like very futuristic. For example, IVF, you have an embryo, and so all the female embryos will have PCDH19 in our case. Would you ever be able to use a treatment like on an embryo or like prior to conceiving?
I mean, theoretically, yes. Yeah, practically, I'm not sure, but theoretically, yes. Yeah.
Thank you.
And I'm curious, in the structural, Paul can comment on this too, with the columnar sort of aggregation of PCH positive, PCH negative, when you look at 10 animals, is there just due to random, when you show some of that, it seemed like there were some areas where the bands were thicker, some were thinner. Is there a lot of variability when you look animal to animal, and could that perhaps in humans, we think this occurs in humans, so we don't know. Could that explain the variability in the phenotype?
Yeah, yeah.
Because this would argue that the synaptic difficulties is at the border zones, right?
Yeah.
Because if you were to somehow able to physiologically study just the cluster of PCH positive and just they should be normal, right? So if you have, I'm just thinking about this broadly. So if you have less, if you have thicker bands, that's going to mean less, less columns that are interfering. So do you see that in the mice?
So indeed, the patterns are random. That's true. And indeed, what we expect is where the borders between the positive and negative cells are located would have changes. So that's why indeed exactly what you said. If you just record from PCD19 positive areas or negative areas, we actually don't really see big changes. But that's why we are examining the patterns of activity in the whole brain using in vivo imaging to really see if the patterns are different. And that's what we expect to see. I mean, the patterns or spread of the activity like you saw in a movie might be different, meaning positive regions and negative regions may not connect with each other, so that spread may stop there. And then rather, there might be more focal activity there that might be leading to a seizure activity or something like those. Is the kind of idea here. But in any case, I think what we need to do is to examine more brain or network level of examination to understand why this abnormal pattern will lead to abnormal brain activity, potentially leading to a seizure activity. And that's exactly what we are trying to do.
Well, thank you. That was a wonderful start.
Thank you very much.